Hello, and welcome to our series, What to Look For, where we talk about specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating Staphylococcus aureus outbreaks. To start, let's go over some of the key characteristics of Staphylococcus aureus, which we will refer to as the abbreviated S. aureus for the remainder of this presentation. Firstly, S. aureus is a bacterium that can produce toxins that cause foodborne illness in humans. These toxins, which are referred to as S. aureus enterotoxins, can only be formed under favorable growth conditions. And importantly, while S. aureus cells are highly sensitive to thermal treatments, such as cook steps or pasteurization, these S. aureus enterotoxins are highly heat stable and cannot be inactivated or destroyed by heat treatments. So what this means in the context of foods is that if you have a raw ingredient that is contaminated with S. aureus, applying an appropriate cook step should be effective at controlling the pathogen. But if a food product is contaminated with S. aureus either after a cook step has been applied or in the absence of a cook step for certain foods such as fresh fruits and vegetables, and is then exposed to favorable growth conditions such as ample access to nutrients, high moisture, and/or temperatures that can allow for S. aureus growth, then this can result in further outgrowth of S. aureus as well as production of S. aureus enterotoxins on food. Subsequently, consumption of food products containing high levels of S. aureus enterotoxin can lead to a type of foodborne illness referred to as foodborne intoxication, which is defined as a foodborne illness that is caused by consuming a food that contains toxins produced by pathogens. S. aureus causes approximately 240,000 cases of foodborne illness in the U.S. each year, and these cases are primarily associated with foodborne intoxication of S. aureus strains that produce enterotoxins. Common symptoms of S. aureus foodborne illness include vomiting, nausea, abdominal cramps, and diarrhea. In general, S. aureus has a short incubation period ranging from 1 to 6 hours, and the illness is typically self-limiting, with symptoms of S. aureus-associated foodborne illness resolving within 24 to 48 hours. Common local establishments where S. aureus outbreaks can occur include restaurants and other eateries, long-term care facilities, schools, community centers, and penitentiary facilities, just to name a few. When investigating S. aureus outbreaks linked to local establishments, there are a few key areas where environmental health investigators should focus their efforts. These include evaluating whether improper food preparation and storage practices, such as improper hot, cold, or room temperature holding practices, as well as improper cooling and reheating practices, could have contributed to the outbreak. Additionally, environmental health investigators may also need to take steps to evaluate whether cross-contamination of S. aureus from food handlers to food products could have contributed to the outbreak. First, let's discuss how to evaluate whether improper food preparation and storage practices could have contributed to the outbreak. In particular, when evaluating whether improper food preparation or storage practices could have contributed to an S. aureus outbreak, environmental health investigators will want to focus in on what practices could have allowed for S. aureus to grow foods that require time and temperature control for safety, or TCS foods abbreviated. This is particularly important given that for S. aureus, it is the toxins produced by S. aureus rather than the organism itself that cause foodborne illness, and TCS foods have specific characteristics that can make them higher risk for growth of pathogens such as S. aureus. For example, an abundance of nutrients, high moisture content, and low acidity or neutral pH. Some examples of TCS foods include meat, poultry, seafood, eggs, various dairy products, cooked or rehydrated grains, and fresh cut fruits and vegetables. To do this, environmental health investigators should identify whether any hot, cold, and room temperature holding practices or cooling or reheating practices used in the establishment were not performed correctly and failed to control S. aureus growth on TCS foods. The goal here is to restrict the amount of time that TCS foods are exposed to temperatures within the temperature danger zone, which represents temperatures between 41 and 135 to 400 Fahrenheit, as these temperatures can potentially support the growth of disease-causing microorganisms such as aureus, as well as subsequent toxin formation on foods. For proper hot holding practices, cooked or reheated TCS foods should be held at temperatures at or above 135 Fahrenheit to prevent S. aureus growth. This can be done by placing cooked or reheated food on preheated tables, chafing dishes, or in slow cookers while serving, or by transporting food to different locations in the establishment via insulated hot food boxes. For proper cold holding practices, refrigerated TCS foods should be held at temperatures at or below 41 Fahrenheit to prevent S. aureus growth. This can be done by storing foods in refrigeration units, walk-in coolers, or freezing units for long-term storage or thawing, or on containers with ice for short-term storage or serving. In general, establishments should limit the amount of time that TCS foods are held at temperatures within the temperature danger zone, which includes allowing foods to sit at room temperature. Specifically, TCS foods should be held for no more than 2 hours at room temperature before serving. Additionally, in cases where the ambient temperature of the establishment exceeds 90 Fahrenheit, TCS foods should be held at room temperature for no more than 1 hour before serving. It is important to ensure that TCS foods pass through the temperature danger zone as quickly as possible, and thus appropriate time and temperature controls should be applied when cooling foods. For example, TCS foods subjected to a cook step should be cooled to 70 Fahrenheit or below within 2 hours, and further completely cooled to 41 Fahrenheit or below within 6 hours. In another example, TCS foods, such as a chopped tomato salad, that are prepared from non-TCS food ingredients held at room temperature, such as a whole uncut tomato, should be cooled to 41 Fahrenheit or below within 4 hours. Similarly, when reheating TCS foods that were previously cold held, for example in refrigeration units, it is important to ensure that the food is reheated to an internal temperature of 165 Fahrenheit for at least 15 seconds within 2 hours after it was removed from cold holding conditions. On the other hand, if food has potentially been subjected to temperature abuse, for example being left out on the counter overnight, then reheating of food should not be performed, and instead the food should be discarded. This is because once formed in a food product due to S. aureus growth, S. aureus enterotoxins are highly heat resistant, and thus reheating is not an effective process for inactivating S. aureus enterotoxins present in food products. When seeking to identify whether any improper food preparation and storage practices could have contributed to an S. aureus outbreak, environmental health investigators should identify whether any hot, cold, and room temperature holding practices or cooling or reheating practices used in the establishment were not effective at controlling S. aureus growth on TCS foods through reviewing relevant records in the establishment. Key records from the establishment that environmental health investigators should aim to review can include 1. Standard operating procedures such as SOPs or recipes, and 2. Documents where monitoring and verification activities are recorded. In particular, when reviewing SOPs, investigators should pay careful attention to hot and cold holding procedures, as well as reheating and cooling procedures to make sure they are appropriate and in line with time and temperature controls that can control the growth of S. aureus on food. Additionally, through reviewing, monitoring, and verification records, the investigator can ensure that SOPs are consistently being followed. For example, investigators should review temperature check logs to identify the frequency in which they are being performed during hot holding on a steam table or during cold holding on ice, as well as logs that show how often thermometers are calibrated and what do calibration records say about the accuracy of the thermometers. Other documents that might be particularly important to review may include checking if cooling and reheating logs show that foods were able to reach the endpoint temperature in an appropriate amount of time. To supplement this review of records, investigators should also conduct interviews with both management and food handlers at the establishment and ask questions about 1. How SOPs for various food preparation and storage practices are enforced, 2. Any potential inconsistencies in SOPs and monitoring or verification records, and 3. Food thermometer calibration and maintenance. Whenever possible, investigators should observe or ask food handlers to demonstrate certain tasks, such as cooling and reheating practices. In addition, when conducting site visits at the establishment, environmental health investigators should closely observe how food is routinely prepared and stored. During these observations, one key thing to take note of is how procedures for hot and cold holding are implemented in the establishment. For example, are steam tables preheated prior to hot holding? Is there sufficient space in refrigeration units or walk-in coolers between food products to allow for proper cold holding? And how frequently are food handlers checking the temperature of foods when serving foods that are temporarily being hot held or cold held? Additionally, environmental health investigators should examine how procedures for reheating and cooling are being implemented in the establishment. For example, are foods being rotated or stirred during the reheating process to ensure even distribution of heat throughout all parts of the food? And is the appropriate equipment being used to facilitate cooling? For instance, in some foods such as soups and stews in large containers, using ice or cooling paddles to steer food may accelerate the cooling process. Finally, environmental health investigators may want to collect select food product or ingredient samples from the establishment and test them for the presence of S. aureus. One key thing to consider when collecting food product samples is ensuring that all collected samples are transported to the lab appropriately. More specifically, it is important to ensure that food product samples collected for S. aureus testing are stored at refrigeration temperatures or lower during transport to the lab. This is particularly important as, according to the CDC's guidelines for confirming the cause of foodborne disease outbreaks, in order for a food product to be implicated as the cause of a S. aureus outbreak, either the food product needs to contain more than 100,000 or 10 to the 5th organisms per gram of food, or S. aureus enterotoxin needs to be detected in the sample, thus ensuring that food samples destined for S. aureus testing are subjected to refrigerated temperature conditions or lower during transport is imperative for preventing possible growth of S. aureus and subsequent toxin formation. Aligned with this, it is also important for environmental health investigators to efficiently communicate and provide instructions for their laboratory partners on what tests should be performed, whether it be tests for the level of S. aureus in the sample, tests of the presence or absence of an S. aureus enterotoxin in the sample, or both. Next, let's discuss how to evaluate if cross-contamination of S. aureus from food handlers to food products could have contributed to the outbreak. It is important to note that some humans can serve as a reservoir for S. aureus. For example, it is estimated that roughly 20 to 30% of humans asymptomatically carry S. aureus in their nasal passages without showing outward symptoms, and sometimes S. aureus can cause skin infections that result in wounds, boils, or rashes. Thus, there is a risk that food handlers can potentially cross-contaminate food products with S. aureus if they aren't frequently washing their hands or wearing gloves when handling certain foods. And in particular, foods that are handled after cooking or require a lot of manual preparation, such as chopped salads, deli sandwiches, or fruit platters, are at high risk for this type of cross-contamination. For example, in a 2015 S. aureus outbreak associated with chantilly cream prepared at a restaurant in Italy, during the outbreak investigation, samples were collected from both 1. various food product samples, as well as 2. the nasal passages of several healthy food handlers. Tests performed on these samples revealed that one sample of chantilly cream showed high levels of the S. aureus outbreak strain at 340 million organisms per gram, and that one nasal swab obtained from a food handler in the restaurant also tested positive for S. aureus and yielded isolates which represented the outbreak strain. Together, these findings suggest that cross-contamination of S. aureus from this asymptomatic food handler to the cream was likely associated with the outbreak. To identify whether cross-contamination of S. aureus from asymptomatic food handlers to food products could have occurred, environmental health investigators should observe how food is routinely handled in the establishment. During these observations, one key thing to take note of is the accessibility of hand-washing sinks and gloves in the establishment to promote frequent hand washing. Are hand-washing sinks properly stocked with soap and paper towels to promote proper hand washing? Are gloves available and easily accessible to food handlers? Additionally, attention should be paid to the behavior of employees with respect to their hand hygiene and glove changing practices. For example, how frequently are employees washing their hands or changing their gloves? Are employees washing their hands after using restrooms? Are food handlers recontaminating their hands and gloves after hand washing by touching their face and other parts of their body? Additionally, investigators can also conduct interviews with both upper management and food handlers about these routine food handling practices. Important questions to ask during these interviews include how is hand washing enforced in the establishment, as well as are food handlers encouraged to wash their hands or change gloves between food handling activities? Are there instances in which food handlers are advised against using hand washing sinks or changing gloves due to scarcity of supplies? These questions are all particularly important to ask, as any practices that deter food handlers or other employees from washing their hands or changing their gloves can potentially contribute to the spread of S. aureus from food handlers to food products. Additionally, when evaluating if cross-contamination of S. aureus from food handlers to food products could have contributed to the outbreak, environmental health investigators may also want to collect select environmental samples from the establishment and test them for the presence of S. aureus. This is because presence of S. aureus on environmental surfaces, especially on food contact surfaces used for food preparation and handling, could provide evidence to support the possible transmission of S. aureus from asymptomatic food handlers to food products. For instance, in the same example described earlier, environmental health investigators also collected and tested several samples from environmental surfaces in the establishment for the presence of S. aureus. And here they found that one environmental swab obtained from a kitchen table surface actually also matched the S. aureus outbreak strain that was found in both a nasal swab obtained from a food handler as well as the cream food product. And thus, here the identification of the outbreak strain on the kitchen table surface provided a clearer link to support that this asymptomatic food handler was likely the source of cross-contamination of S. aureus to the chantilly cream in this outbreak. Key things to consider when collecting environmental samples includes making sure that the right tools are used to collect samples. For example, using sponge kits with sterile gloves or sponges on sticks are typically preferable to using cotton swabs, as they can cover a wider surface area and capture more environmental microbes. Additionally, in some cases, investigators may have to disassemble certain types of equipment, such as this deli slicer, in order to access hard-to-reach sites inside the equipment that could potentially be harboring S. aureus. This may require additional support from maintenance personnel at the establishment to properly break down the equipment. In summary, in this presentation we have covered some of the specific characteristics of S. aureus as well as several key areas that are relevant for environmental health investigators to consider when investigating S. aureus outbreaks linked to local establishments. In addition to Staphylococcus aureus, check out our other videos that cover key considerations for investigating foodborne illness outbreaks linked to other foodborne pathogens.